Yeah, the title of my talk is The Body Parcellates the Brain. And I would like to first start by saying what do I actually mean by the body and the brain, and will then proceed by explaining what I mean by saying that the body parcellates the brain. The representation of the body in the brain has actually quite long history, and seminal work has been conducted by Penfield and colleagues, showing that stimulating different parts of sensory motor cortex results in distinct and clearly separable sensations, but also movements at different parts of the hand. Whereas stimulating a bit more inferior parts of sensory motor cortex results in distinct sensations, but also movements at different parts of the face. Similar work, and also work including modern imaging technology, has led to the concept of the so-called homunculus, which kind of describes a one-to-one relationship between the physical outline of the body and a map-like representation in the brain. The so-called homunculus-like representations, or one may also call them somatotopic maps, are perhaps the main functional principle to understand the function of sensory in motor cortices. However, when you look at currently used parcellation schemes, such as, for example, the one proposed by Brodmann, you realize that such somatotopic representations or homunculus-like structures cannot be found in those kind of areal delineations. For instance, when you look at Brodmann area 4, which is primary motor cortex, or you look at Brodmann area 3, which is primary somatosensory cortex, you realize that both areas are quite homogeneous, and there is no kind of sign of body part-specific areal delineations. When you accept the view that cortical parcellation schemes do provide us with the basic organizational principles of the human brain, then you may conclude that perhaps body part-specific processing is not a general organizational principle of the human brain. However, by the time there were also other researchers proposing cortical parcellation schemes, such as Paul Flechsig. Paul Flechsig used changes in cortical maloarchitecture over time to propose a cortical parcellation atlas. Other than Brodmann, he actually did distinguish different fields within primary motor cortex and within primary somatosensory cortex based on different maloarchitectonic changes he detected within those two fields. He was assuming that perhaps those distinct fields could perhaps relate to different representations of the body. However, he couldn't really test his assumptions. This might remind us a little bit of so-called septa. I'm not sure who of you are familiar with the concept, but septa are myelin-poor boundaries that have been detected in monkeys and mice, and they form boundaries between adjacent body part representations. Here you see the so-called hand-face septum, which is a myelin-poor area in the input layer 4 of primary somatosensory cortex. However, neither the kind of somatotopic parcellation idea by Flechsig nor the existence of septa in humans have so far been identified. To kind of approach that goal, the study had 3 aims. First, I wanted to show that there are myelin-poor structures within primary motor cortex and within primary somatosensory cortex that form or separate the areas into distinct fields using in vivo cortical myelin mapping. Secondly, I wanted to show that there is a correspondence between those local changes in cortical maloarchitecture and the somatotopic representation of the body in both areas. And finally, to kind of pursue a multimodal parcellation approach, I aim to show that those local changes in cortical maloarchitecture are also related to changes in the underlying functional connectivity network as measured during the resting state. To start off with, what you see here is a cortical myelin map of one single participant measured with 7 Tesla MRI. Myelin is here displayed by means of a quantitative T1 signal measured in milliseconds that was acquired using a MP2 range sequence of an isotropic resolution of 0.5 millimeters. What you see is that there are clear boundaries within those kind of myeloarchitectonic pattern within primary somatosensory cortex. And when you look a bit more closely, you see that there is a clear boundary that kind of separates the primary somatosensory cortex into more inferior and more superior parts. Now, the same participant also underwent functional imaging, and we asked the person to move the fingers just like that, or to move the lip. And I could show that basically those structural myelin boundary does correspond to the functional boundary between the hand area and the face area. The same was actually true for primary motor cortex, where there were also clear boundaries within the area corresponding to the functional boundary between the hand and the face. Now that we had this great and very high resolution, I think, of 0.5 millimeter isotropic, I also had a look at layer-dependent changes. And what I think is interesting, what you see here is actually cortical myelination values averaged over the 7 subjects I measured with the 7 Tesla in the peak of the hand area, in the functional hand-face border, and then the peak of the face area. And what you see that the myelin reductions are significant in more superior and inferior cortical layers in primary motor cortex, but are significant in the middle cortical layers in primary somatosensory cortex. Whereas this is still preliminary. I think what it shows is a very interesting specification within primary somatosensory cortex to the middle cortical layers, which is somehow reminding us of the specific myelin reductions in the input layer 4 of septa that I showed you before. We then were interested whether we could generalize our findings to a larger population, and we used the data as provided by the Human Connectome Project for that purpose. Myelin is here defined by means of a T1/T2 ratio. And what you again see when you just visually inspect those data sets, it's an average over more than 400 participants, is that there are inhomogeneities, again, in the myeloarchitecture in primary somatosensory and primary motor cortex. When we then sampled values from superior to inferior, just parallel to the central sulcus, we indeed found that this myelin reduction is statistically significant. We then, within the same subject sample, had to look at the functional hand-face border. We saw again that there's a correspondence between the myeloarchitecture and the functional border between the hand and the face. And when again sampling values along those same 2 cortical paths I was introducing before, then we found that the reduction of cortical myelin between the hand and the face was very much corresponding to the functional border between the hand and the face in both of those 2 areas. Finally, to pursue a multimodal parcellation approach, I was interested whether there would be a correspondence between local changes in cortical myeloarchitecture and the underlying resting state functional connectivity networks. For this purpose, we were taking seed regions in the highly myelinated hand area and in the highly myelinated face area, and we were finding a kind of strongly correlated hand network across the sensory motor boundary, and a kind of strongly correlated face network across the sensory motor boundary, but not so much crosstalk within those 2 areas. When we then took as seed region the kind of poorly myelinated gap between the hand and the face, then we saw that the resting state functional connectivity was quite low, so was quite minimal between those 2 areas. Those analysis and also similar analysis that you can read in the full publication that goes with this study, we actually show that there is a correspondence between the local myelin reduction between the hand and the face representations and the diversion of the underlying resting state networks between those 2 body part representations. So to sum up, I think what the study shows is there is a kind of patchy myeloarchitecture in primary somatosensory and motor cortex that may be used to differentiate different kind of subfields within those areas. Those subfields could be related to the functional representation of the hand and the face. Or by using layer-dependent analysis, it could also be shown that there is in my view, very interesting specific reduction within the middle cortical layers of primary somatosensory cortex with respect to the myelin reduction, which may indicate, I think much more research has to be done, that that this may be a homologue of so-called septa, as have been identified in monkeys and mice. We validated the finding using a larger subject sample, both the functional and the structural findings. And finally, we found correspondence between the local variations in cortical myelin and resting state signal fluctuations in those 2 areas. So what do we take from this? And I think this will be my final slide. I think, first of all, one may think that even though regarding primary motor cortex and primary somatosensory cortex as homogeneous areas is a kind of very deeply rooted concept for us, one may rather perhaps think about a motor output hand area, a motor output face area, a motor sensory input hand area, and a sensory input face area. So in a way, regarding those distinct areas, it's also functionally distinct areas. And in particular, I think it's interesting that within primary motor cortex and within primary somatosensory cortex, you have those borders between the body part representations being deeply rooted in changes in cortical microstructure. And I think a very kind of obvious implication is for models and studies on cortical plasticity, for example, where you study the rearrangement of somatotopic maps over time, for example, or with learning, or even after de-differentiation. I think for those studies, it's very important to think about those fixed structural borders. However, I would like to take my last 30 seconds to point out even something different or something additional. I think it is important to know or to realize that we don't only have somatotopic maps in sensory and motor cortices. There are many, many more those somatotopic maps within the brain, such as in parietal cortex, even superior parietal cortex, or, for example, in the superior occipital cortex. And I mean, there are many, many more areas that show hand and face specific processing, not only cortical areas across the hierarchies, but even subcortical areas. And therefore, it's important to realize that those hand-specific networks and the face-specific networks that you do find across the cortical hierarchy and that are manifested by changes in microstructure, on the other hand, in primary sensory areas, may actually let us think about how many of the cognitive and sensory motor and cortical computations realized by cortical areas do actually relate to those specific hand and face selective networks. For example, when we detect during language production activity changes in the superior parietal cortex, we might not so much think about whether it's the hand area or it's the face area of superior parietal cortex. We just may say, okay, it's superior parietal cortex. But we may perhaps a little bit more think about the body part-specific processes that may relate to many of the functions associated with the hand, such as tool use or gesturing, or with the face, such as language production. And finally, I think, in my view, showing that body part-specific processing is associated with changes in cortical microstructure is another very nice and important evidence that how embodied actually the human brain and perhaps also the human mind might be. With this, I would like to thank my collaborators for their great contribution. And I would also like to thank you for your attention.
The floor is now open for questions.
I mean, so somatotomy is quite a complex thing. I mean, I just wonder, why do you find this border only between hand and face and nowhere else along the motor homunculus?
Yeah. Thanks a lot. I think that's a very, very interesting question, and it's just a matter of time that I didn't go into that. So Flechsig originally proposed 3 differentiations. So he said there is a lower third, there is a middle third, and an upper third within both areas. So it's very, and also when you look at the parcellation atlas just recently published by Van Essen and colleagues, you realize that they also distinguish more than just the hand and the face areas, but they distinguish also the leg area additionally. So in my view, it is very likely that they also have a separate representation of the legs, for instance. I just didn't test it with my data so far. Thanks.
Carl?
I personally doubt that this is a microstructural finding because you have a spatial resolution of 0.5 millimeter. 0.5 millimeter is a lot regarding the ending of myelinated fibers in the cortex. So at some places you underestimate the myelin content, especially in the sulci. At other places you may overestimate the myelin content, if this is myelin at all, what you are measuring. And when you look at the border between hand and face area, you have the omega structure of the hand region, and then you have a sulcal indentation to the face area. So what you find is the sulcal pattern. And this sulcal pattern is reflected by different depth of the myelin within the cortex. So the myelin is not the primary structural, in my opinion, is not the primary structural effect. It's the sulcal indentation at this point, and therefore you find it here and not everywhere.
Mm-hmm. Okay, I think that's a complex question. I think I could talk for half an hour to answer the different sub-questions. I will start with one thing. So the signal we used, I think it's important that we used a validated signal for cortical myelin. So the quantitative T1 signal has been validated by Carsten Stüber. So at least 2/3 of the variations of the signal may be related to myelin. That's one point. Another point, I think it's important to point out that we used the MyPath framework, which has spent very much effort to model the curvature in a kind of biological sensible way, so much better than FreeSurfer, for example. So even though I agree that there may be changes in the myeloarchitecture related to changes in curvature, the framework does at least account for some of the variations. I also think it's worth mentioning that, I mean, the border kind of extends through different so I looked at single subject data, so I looked at really the structure subject by subject, and it's not that the curvature is, I see changes in the curvature in each subject, but I also see this additional change related to the border. And sometimes this change is really inhomogeneous, so it has different shapes, and it always kind of follows that fine-grained structure. So in my view, that's kind of a nice single subject validation of those findings. Additional question? Do you want to answer? Yeah.
I did measurement of myelin in myelinated, myelin-stained sections, and I cannot find such a decrease in myelin.
Yeah, the thing is, one additional comment on this might be that it's difficult to, I mean, when you look at the structural changes, it is very difficult to, just by looking at the structure, you wouldn't actually say this is a clear drop. So you only see it in combination with the function. Yeah, so that's, in my view, difficult only with I mean, I totally agree that post-mortem validation is the gold standard, and you should always combine this with post-mortem studies. I just see the difficulty that only looking at post-mortem data, it's very difficult to know where exactly you should look at. I mean, in my view, that's a difficulty. Yeah. But thanks a lot. I totally agree that one should have a look at the post-mortem data.